Hey there folks, and welcome to video 3, the last video, for Chapter 10, the molecular biology of the gene. So we have been looking at how our DNA is built, and how we make more DNA, and how our cells read off that genetic information in the DNA so that they can actually be used. Not all organisms keep their DNA to themselves, however. Some like to spread it around, usually in ways that damage those other organisms. Specifically in the case of viruses. Viruses are terribly simple. They're just protein shells around DNA. Some of them aren't even that. They're just protein around RNA. But they're so simple, they literally don't have the hardware required to reproduce their own DNA. This is why they infect us and everything else. And they hijack our cells' natural cell cycles to make new DNA for them and to reproduce the virus for it. Viruses that specifically and only infect bacteria are called bacteriophages, or sometimes just phage for short. When this happens, sometimes the virus breaks out quickly once the bacteria has done the job for it, and sometimes the virus DNA hangs around inside the bacteria for a long time. This forms two different life cycles for viruses. The lytic cycle, here on the left-hand side of this diagram, shows you where a phage first infects a bacteria cell. It injects its DNA into the bacteria, and that bacteria DNA circularizes it. It forms its own little bundle. Certain factors inside the bacteria then determine whether or not the bacteria DNA stays in the lytic cycle or enters the lysogenic cycle. If the DNA stays in the lytic cycle, it will be reproduced. That DNA will be reproduced along with the bacteria's DNA the next time the bacteria goes through reproduction. And it's going to make a whole bunch more little viruses. The new phage DNA and its proteins are synthesized, they're assembled into new copies of the virus, and then those viruses break free, physically busting the bacteria cell open. And that spot right here where the cell lyses, releasing the phages, this is where we get damage due to viruses. It's that stage right there where inevitably those brand new viruses have to get out and they start destroying cells. If conditions are right, and instead of falling into the lytic cycle, the phage DNA will fall into the lysogenic cycle, which means it's going to hang around for a while. And instead of circularizing and staying that way until it reproduces, the phage DNA integrates itself into the bacteria's DNA, becoming what is known as a prophage. And that DNA gets reproduced along with the rest of the bacteria's DNA still attached, hidden inside the bacteria's DNA, sometimes through many different rounds of transition, sometimes staying there for a long time, being passed down from daughter cell to daughter cell in the bacteria's DNA. And eventually, occasionally, the prophage will exit the lysogenic cycle, enter the lytic cycle, and then we have exploding cells all over the place. Viruses, unfortunately, don't just attack bacteria. Sometimes they attack us. And this is when they cause diseases in animals and plants. They can follow lytic and lysogenic cycles in animals and plants just like they do in bacteria. It's the exact same thing. But instead of being in a bacterial cell, they're in our eukaryotic cells. The protein capsid around the viral DNA or RNA, depending on what type of virus it is, enters the host cell. This is what the infection is, and adds the viral genome to the local DNA. And depending on whether or not that virus is following the lytic or lysogenic cycle, those new cells, those host cells, will either end up making new copies of the virus right away, lysing the cell, or at least damaging the cell as they leave, or they'll hang around for a while before that happens. Because of this behavior, there are some viruses that can severely threaten human health. Things like the Ebola virus, which can be very deadly. We're much better at treating Ebola, by the way, than we used to be, and that's very new technology. Some viruses, however, continue to be a pain to our existence no matter what we do, because there are tons of different versions of them. There are really only a handful of versions of Ebola, which is why we have gotten so much better at treating Ebola patients so quickly. But there are tons of versions of the virus that causes the common cold, and that's why it's so very difficult to keep up with. It's hard to predict what new mutations the virus might come up with. And treating it, well, it's a catch-up game. We just try and make having a cold as bearable as possible. But still, because viruses can change so quickly, some of them are and remain terrifically dangerous. We're much better at treating Ebola than we used to be, but it's still one deadly virus. So is HIV. HIV is the virus that causes AIDS. And again, AIDS patients have much longer life after infection than they have ever had before. And they have much better quality of life than they have ever had before. The difficulty with treating AIDS and HIV patients as well as we need to in medicine is partially because of the side effects that the virus has, and partially because of how it reproduces. Most viruses come in and they inject their DNA. However, HIV is what is called a retrovirus. It injects its RNA and works backward to create its DNA inside our host cells. And the difference in that process means treating it, let alone preventing it or treating the symptoms, can be a little bit more complicated. But again, our medical technology for treating both retroviruses like HIV and very fast-moving viruses that with really severe side effects like the Ebola virus have much improved, especially in the last 20 to 30 years. Right now we are alive at a time period where our ability to save lives that otherwise would be horribly affected and ultimately ended by these viruses is vastly improving. And those patients that contract Ebola or HIV infections live much longer and may recover, especially in the case of Ebola, or live a much longer time and have a much better life post-infection in the case of HIV. Viruses are not the only things that can infect our cells. Plants can be infected by infectious forms of RNA called viroids that are smaller and simpler and harder to fight than viruses. And mammals can be infected by something that is even smaller still. It's not even infectious RNA. It's just a kind of infectious protein called a prion. So there's a host of other things, it's not just viruses, that can infect both prokaryotic and eukaryotic cells. Some things just can't keep their DNA or their reproductive cycles to themselves, usually because they are so simple they need to hijack another cell with the correct hardware in order to make more of themselves. Some living things, however, can use these abilities to their advantage. Bacteria are incredibly good at this. Bacteria are able to receive new DNA, either from their environment, from a phage, or from another bacteria, and add to their own DNA, adjusting it over their life cycle. Imagine if we could do this. This is how bacteria become resistant to our antibiotic medicines. When they encounter samples of the antibiotic in their environment, eventually those that survive end up having antibiotic resistant genes. it is possible for existing DNA to pick up those antibiotic-resistant genes from their environment when the old bacteria die or donate it out into their environment. And those recipient cells, they now have the antibiotic-resistant gene. By the way, this is why it's so important for patients to always finish their rounds of antibiotics. If we stop taking the antibiotics early, just because we feel better, as we often do before we're done taking all the antibiotics our doctor gives us, it leaves some bacteria from our infection behind. And those that have survived so far are frequently the resistant cells. It means that we can accidentally breed our own antibiotic-resistant bacteria. And that becomes a real problem later on when we need to treat that particular bacteria infection later. It becomes harder and harder to do it well. That's called bacterial transformation, by the way, when a bacteria can pick up new DNA sections from a donor cell that releases it out into the environment. When bacteria become infected by a phage, this is called bacterial transduction. And the release of the phage from an infected donor cell leads to a new round of infection. And remember those lytic and lysogenic cell cycles we were just talking about? That recipient cell that ends up integrating the phage DNA into its DNA is undergoing something called bacterial transduction, where the DNA from the phage is being brought into the bacteria DNA. It's not from another bacteria. If it was from another bacteria and was brought in from the environment, it would be transforming the new bacteria's DNA. But because it's coming from outside, not just the DNA, but from not just the bacteria, but from a completely different kind of organism, from a virus, this is what makes it transduction. When bacteria acquire new proteins and new DNA in the form of plasmids, those round sections of DNA that can reproduce separately inside a bacteria, this is called bacterial conjugation. A section of one bacteria's DNA can be coded into a plasmid, sort of a little carrier, a little messenger form of DNA that can be pushed across a bridge that two bacteria build between themselves. That will move from the donor cell to the recipient cell. And once the plasmid reaches the other side of the bridge, it can break open, and the recipient cell can integrate that one specific transposon piece of DNA into its DNA. We can do this in labs, and we also see it happening in the wild. Plasmids, like you see at the top, can then be reproduced through various rounds of cell replication in the wild. And those plasmids can also be integrated straight into the bacteria's DNA, as you can see at the bottom, and then be reproduced through various rounds of cell replication inside the bacteria's DNA. When we use DNA technology to get bacteria cells to make important hormones and proteins for humans to use in medicine, things like estrogen, testosterone, and insulin, we are frequently using bacteria's ability to do this exact thing to get it to start secreting human proteins and hormones by injecting plasmids with the DNA for making those proteins and hormones into the bacteria so that they can be integrated, and then those daughter cells, through cell replication, will make the hormones and proteins for us. Then we can harvest them from the bacteria, purify the hormones and proteins, and they'll be ready to help human patients. Alright folks, this is going to wrap it up for the end of Chapter 10. I know that middle section there was awfully technical, but you made it through. Congratulations, guys, and I'll see you next time. Bye.